I'm Olivia Archambault.
I'm John Minderman.
I'm Elizabeth Dudley.
I'm Mackenzie Tilley.
And I'm Russell Bowles. This is our project on the acquisition of photosynthetic chloroplasts from algae in Elysia, chlorotica.
Horizontal gene transfer, HGT, is a mechanism of gene transferal that is independent of sexual reproduction, whereas vertical transfer occurs when genetic material is passed from one generation to the next. This image depicts the evolutionary scheme for primary, secondary, and tertiary plastids. The secondary endosymbiotic origin of plastids is illustrated in Vipera litoria from the red algal lineage. The subsequent acquisition of V. litoria plastids by the sea slug Elysia chlorotica in a tertiary endosymbiotic event imparts photosynthetic activity to the heterotroph. Photosynthesis occurs in chlorophyll-containing plants, algae, and some bacteria. Some species in the aquatic phyla Mollusca, Porifera, Cnidaria, Acelomorph, and Chordata have evolved to capture photosynthetic products such as energy and carbon through symbiotic relationships with algae or cyanobacteria. The photosynthetic abilities of the species derived from the consumption of algae or cyanobacteria is not permanent. It is a horizontally transmitted gene. Each generation acquires its own photosynthetic partner. Captured plastids, or chloroplasts, are retained intracellularly in cells lining the digestive tract of sea slugs. They provide the slugs with energy and carbon. These organisms have not evolved to pass on the ability to photosynthesize to their offspring, but have evolved to help retain the plastids and stay photosynthetic once the chloroplast is obtained. It is possible that the sea slugs can retain the photosynthetic chloroplast for the entirety of their lives, or 10 to 12 months. Results suggest that algal nuclear genes are present in the sea slugs and that plastid protein and pigment turnover, which are necessary to sustain powers of photosynthesis, take place in the cells of E. chlorotica. However, it is not known if the genes of the algae have been directly passed on to the sea slugs. Multiple studies have been conducted to test whether or not the sea slugs derive genes from the consumed plastids, but all have been largely inconclusive. Due to errors in these experiments, the data cannot be fully trusted. If transferred at all, it is understood that algal nuclear cells will be of a very low copy number, and their expression will be low.
Several experiments were set out to see if E. chlorotica receives genes from algae to become photosynthetic. They explored whether nuclear gene transfer occurred or just plastid transfer from the algae to E. chlorotica.
The researchers discovered that chloroplast-encoded sequences were present in the RNA of the sea slugs. 101 chloroplast-encoded protein-coding genes matched sequences in the algae. 60 were in contigs, or a set of overlapping DNA sequences, and 41 were in rod reads. 36 sequences which mapped to ribosomal protein subunits matched between slug and algal RNA and with chloroplast genome sequences.
88 out of 111 slug transcripts in the chloroplast-encoded datasets were identical. What this means is that many of the DNA and RNA sequences found in the algae and the sea slugs were identical. In order to prevent any contamination in the experiment, the researchers starved the slugs for 2 months to ensure that no algae was mistaken for E. chlorotica matter.
The data shows that the chloroplast genome is active within E. chlorotica cells and is producing transcripts for some chloroplast proteins. Several transcripts found code for components of photosynthesis 1 and photosynthesis 2 reaction centers. CYTF and Rubisco transcripts inside the slug show that the E. chlorotica synthesized these proteins. This indicates long-term integration of plastid function in the slug cells. However, the biochemical mechanisms for this integration is not yet known.
In the multiple experiments conducted, there were multiple hypotheses. The experiments conducted by Palaciu, Wagale, and Pierce, respectively, hypothesized the following: that a combination of unusual plastid stability, horizontal gene transfer, and long-term maintenance of cryptic algal products maintained the plastid cells. That there is an incredible plastid longevity, but no nuclear gene transfer because the feeding process, no nuclear genes are transferred to maintain the plastid cells. That the nuclear and chloroplast genes were transferred to produce plastids and proteins in the slug. Problems with the first two experiments include the following. They assumed that there would be a large chloroplast transcript presence in the slug cells. as in the plant cells, and would therefore be easy to identify. This assumption is unproven and unfounded, and therefore is most likely false. RNA taken from the slugs are from slugs that did not go through photosynthesis, and therefore most likely didn't go through gene transfer to make the plastids. The machines and techniques used only examined a fraction of the genes, and the database they compared it to didn't have the algae the plastids most likely came from. Both experiments make conclusions, either of which may be correct, but neither had enough data to support their conclusions. In conclusion, the first two experiments uses PCR techniques. They returned fewer number of genes. Though more accurate, they found less genes and were able to compare less. Because of the size of the DNA and protein sets, it is easy to find conclusions from this limited data, but to prove the solution requires much more sequencing to prove. Therefore, no correct answer is present, and the data is still inconclusive until much more sequencing is done.